Yeah, so my name's Elizabeth Hinde and I'm in the School of Physics here at the University of Melbourne. And today I'm going to tell you about fluorescence and how it can be used to observe DNA in a living cell. I thought I'd just start by telling you what I did after I did high school physics. So this is a picture of my dad, who was actually my physics teacher, so that's why I selected physics in year 11 and year 12. And then after that, as a result of taking physics and maths, I enrolled in a Bachelor of Science at ANU in Canberra, where I majored in physics and chemistry, and that's where I first learnt about fluorescence and became interested in it. And then after that, I went and moved to Melbourne and studied. I departed from science or traditional science and did a master's in painting conservation, which again still came back to being related to fluorescence, and I'll tell you more about that. And then after that, I went and did a postdoc in California with Enrico Graton, shown here, who's an expert in fluorescence microscopy. So I first learned about fluorescence, I guess, in my Bachelor of Science, but it became a theme. It certainly directed me towards my PhD project and then ultimately a postdoc in America. And so now at the University of Melbourne, I'm continuing my research in fluorescence, but now looking at biological applications. And so we have a molecular biology lab, and we use a lot of fluorescence microscopy to study cell biology, and in particular how the genome is organized. So today I'm going to tell you first about what is fluorescence, and then I'll talk about how fluorescence can be used in biomedical research to understand cell biology. So what is fluorescence? Fluorescence is the emission of light by an atom, molecule, or material that has absorbed electromagnetic radiation, and examples are minerals, many marine creatures. So for example, genetically encoded fluorescent proteins came from a jellyfish initially. And it's also observed a lot in many types of pigments. And so if you went to the NGV recently, you might have seen the Keith Haring exhibition, and they had a room lit by ultraviolet light so that you could see the fluorescence from these paintings. And so fluorescence is a form of luminescence that ceases almost immediately upon removal of a radiation source. And so this is unlike phosphorescence, which you may have also seen, like in the form of glow-in-the-dark stars, which persists for some time after you remove the source of radiation. So what is fluorescence at the level of atoms? Fluorescence occurs when an excited atom or molecule relaxes to a lower energy state through immediate emission of a photon. So this is the quantum of electromagnetic radiation. And it depends on the electronic structure of the atom. So here we have a schematic of an atom, which you may have seen before. You have the nucleus, and then these electronic shells that are organized according to their energy level. So as you go out further, there's an increase in the energy level. And essentially, these electron energy levels, each shell is associated with this specific amount of energy, and electronic transitions between these shells are possible when an atom absorbs a photon of light. So upon absorption of a photon of a particular amount of energy, this transition can take place, and it can be excited to an excited energy level. And then what happens next depends on the atom. So in some instances, upon absorption of a photon, the electron will be excited to a higher energy level, and upon relaxation, there'll be an emission of another photon. And the relaxation from this excited state and the immediate emission of a photon that has a frequency and a wavelength dependent on the energy difference between these two levels is what we call fluorescence. So this is fluorescence at the level of atoms, and we have a couple of demonstrations of this. So just to demonstrate what fluorescence looks like in terms of atoms before we move on to molecules. So I'm not sure how to turn on the gas or do this part.
You got to press that safety valve first.
That one?
Yeah, this one here.
Okay.
Turn it on.
Hang on.
Then press up the mode.
Okay. So like here we have different like molecules or which contain atoms that should undergo, like be able to have this excitation of an electron to a higher energy state.
How do we know what's going to happen? Like, how do we know that, what are we looking for?
We're looking for an emission of a photon, potentially like a different color depending on the energy difference in the energy levels of that atom. So you can imagine, because wavelength is inversely proportional to the energy state, and wavelength is the indicator of the color, you would expect that depending on the atom, there should be a different type of color associated with that electronic transition. So here we have copper sulfate. So that's like a green.
All right. Cool. So is this why copper spires on old churches are green?
Potentially.
Potentially. Oh, I'm asking tricky questions. All right. Can we play a game? What have you got next?
Oh yeah, so maybe like remember this was green, so consider which one maybe has a higher difference in the energy level, like if it's inversely proportional to wavelength. So that was like a green fluorescence, and here we have lithium chloride.
All right, but before we spray, what color? Any guesses? Yellow. We've got yellow, we've got red from the audience. I can't see the chat at the moment. Can you see the chat, Benita? Has anyone put up there? No one's put anything up. All right, we've got yellow, red. Any other colours? You're going with red? Okay.
Oh,
Nice.
yes.
Well done. that one sticks around for a while.
Oh yeah, it's true. Yeah, that's a good. And then we have sodium chloride.
Ooh, colours. Sodium chloride, I know. What's that? Table salt. Nice. You can see the chemistry people in here as well. Cool. What colour? Blue, orange. Okay.
Ooh.
Orange, yeah.
So you can see that just from the fluorescence emission that you can obtain some information about the atom or the matter that you're trying to interrogate. And essentially that's like the principle of spectroscopy, is observing the interaction of some, or usually electromagnetic radiation with some form of matter.
All right, so you were going to ask which ones required the most energy to create the light or the fluorescing.
Yes.
So we had copper was green. Should we put them in like, maybe
Yeah.
I'll turn the file.
So yeah, copper was green. Lithium chloride was red, and sodium was yellow. So yellow, so green being the lowest wavelength, yellow intermediate, red longest wavelength.
Yeah, so Liz gave you a hint before. Which one would require the most amount of energy?
And you have here's the relationship of wavelength with respect to the difference in energy between the different energy levels.
Big wavelength. Big or small energy? Small. Small. Yeah, inversely proportional. Cool. All right.
Yeah. Okay, so that's in the context of atoms, but with molecules, it's a bit more complicated. So if you keep in mind this diagram, which is called a Jablonski diagram, you might have come across this in chemistry. So if we move on to molecules, which are essentially made up of multiple atoms that vibrate, the energy associated with the vibrations is actually superimposed over those electronic energy levels that we were discussing before. So instead of just considering does an electron get excited to some excited state as a result of undergoing a transition between these electronic states, in the case of molecules, because you have these vibrational energy levels superimposed over the electronic energy states, there's far more transitions that can take place. So in the case of an atom, when it absorbs in a photon, if that atom is capable of fluorescence, then the photon that it will emit usually has exactly the same wavelength. And this is because there's no vibronic energy structure. But in the case of a molecule, the molecules upon excitation from this ground state to some excited state, which can be the energy level 1 or energy level 2, they will rapidly relax to the lowest vibrational state within a given energy level prior to emission. And because of this loss of energy, which we call internal conversion, the photon that is absorbed is no longer exactly equal to the photon that's emitted as the fluorescence. And what you see is something we call the Stokes Shift, which you'll see later in the talk is very useful actually. The photons that are absorbed in the case of molecules generally result in emission photons that are at a longer wavelength. So there's always this shift in terms of the absorption spectrum versus the emission spectrum of a molecule that fluoresces, as opposed to an atom where it has this resonance fluorescence, because the photon that's absorbed is approximately equal to the photon that's emitted. So this brings us to fluorescence spectroscopy, which is the study of the interaction of electromagnetic radiation with fluorescent materials, atoms or molecules, that are capable of undergoing this fluorescent process. And to really look at where an atom or molecule absorbs electromagnetic radiation and to understand what is the wavelength of the photons that it emits, we use something called a fluorometer. And so essentially this is an instrument that enables us to look at what wavelengths a fluorophore absorbs and what wavelengths a fluorophore emits so that we can study that molecule and detect it. So for example, if I took a series of fluorescent pigments and dissolved them in something that made them into a solution, on this instrument I'd be able to record their emission spectrum, which is where I would excite this fluorophore with some wavelength and then monitor the intensity of fluorescence at all the possible wavelengths and obtain spectra that looks like this. So you can see here with the excitation spectrum, for each of these pigments, I now know what wavelengths. You can see they all have a peak around here and here. I can see what wavelengths they absorb at. And if I excite them at this wavelength, I can see at what wavelengths do they emit. So I can start to understand how can I make that atom or molecule fluoresce. And if it does fluoresce, how do I detect it? And so by recording their excitation emission spectrum, I can start to understand how I might try and detect this molecule, or if I don't know what the molecule is, it might give me a clue as to what it is. As you saw here, you could tell from the color maybe what atom you were looking at. So an application of fluorescence spectroscopy is looking at fluorescent pigments, as I've alluded to in the previous slide. So daylight fluorescent pigments have been adopted by several significant artists. For example, Keith Haring, which I mentioned before, that recently had an exhibition at the NGV. But also you might have seen paintings by Howard Arkley, who was a Melbourne artist who depicted suburbia. And essentially, these fluorescent pigments are quite useful to people who work in a gallery or an art curator, because it gives you a clue as to what pigments they use. It also gives you a way to monitor their deterioration. And so UV fluorescence photography is often used actually to look at traditional pigments, like from Renaissance artists, to see if it's a fraud or not, depending on the fluorescence signature. But in the case of these modern paintings by like Howard Arkley or Keith Haring, there was no fluorescence photography set up to selectively look at what are called daylight fluorescent pigments. So as their name suggests, rather than being excited in the UV region, they're excited by visible wavelengths. And so we wanted to establish a photography setup specifically to extract the visible fluorescence from paintings. So here we have the idea, like if you have your painting set up here, if we know what wavelengths the pigments in the painting are excited at, and I've told you that they're visible daylight fluorescent pigments, so they're actually excited around 450 to 500 nanometers. And if we know where they emit, so you can see here the emission spectra, and you can see it emit between 450 and 650 nanometers, we can set up a filter system where we put our source of light, a filter in front of it that exclusively emits light at the wavelength that they're excited at. And then in front of the detector, or the camera in this case, we put a second filter that selectively selects these emission wavelengths and blocks any reflection from the excitation source on the painting. And so when we do this, here we have a work by an artist called Ry Hastings, where you can see from just a regular photograph, it's hard to say if any of the pigments in this painting are actually fluorescent. And here's another painting you might have seen at the Ian Potter Museum by Robert Rooney, where again, it's not really clear from a regular photograph if any of the pigments are fluorescent. But if you use the daylight fluorescence setup, you can see where the pigments are actually fluorescent. And so this gives art curators a way to document the painting, also how to monitor how it's fading as a function of time. And importantly, because the artist used a fluorescent pigment, it's important from a point of view of display to include the wavelengths it requires to fluoresce so that the artist's intent isn't lost. So this is an example of how you might use fluorescence emission and excitation spectra in like a real world setting. So that completes this part, which is focused on what is fluorescence, but just to reiterate that some things are ultraviolet fluorescent versus visibly fluorescent, we have another demonstration.
Really close. All right. Okay, I'll get you to stand close to me, Liz, so I can use your microphone as well.
Oh, yeah.
So I went through my house with what we call a black light. Do you want to explain what that is?
It's a source of ultraviolet radiation. So that's pretty good. Like, if you can see, actually, that's another good point. Like, sometimes it doesn't matter. The emission spectrum will be the same irrespective of the excitation you use, so long as it's sufficient to cause the transition in the electron going to a higher energy state. So ultraviolet's a good source for many of the like visible types of fluorescence because it's a shorter wavelength, and so it ensures that you'll achieve the excitation for fluorescence to occur.
Cool. So I have got, hopefully you can see on the camera there, I've got an egg. Don't even know how I got it in one piece on the way here in my car, but I did. I do a bit of sewing, and I just bought in a whole bunch of things, some flowers, different types of laundry detergent, okay, tonic water. I know you're all too young yet, but who likes a gin and tonic? You don't have to say. And some olive oil, always tasty as well. Thread, all sorts of things. So, oh yeah, and my lunch, my leftovers from lunch, okay, banana, why would I do that? So, oh yeah, and some money and my credit card. All right, so we're going to have a look at these objects and see which ones fluoresce under UV light. Now, if we were to turn the lights off in here and it was completely black and so there were no light sources, how many of these things do you think you would be able to see? Any? Without UV. I mean, they're not these things aren't fluorescing at the moment, are they? They're reflecting light, and that's why we can see them. So if we were to turn all the light sources off, we wouldn't see them. All right, I'm going to pour in some, because this makes it so much more effective. Pour in some of the tonic water. And some olive oil. Yeah, you're welcome to. No, we don't eat food in physics because we never know when there's going to be radiation around and all sorts of things. And Liz has just been spraying chemicals all over the place, so, you know. All right, any guesses as to how many of these things are going to fluoresce? Sorry? A couple? Okay, all right. What about the egg? Like, you don't want to take a punt. Okay, why would I bring in washing detergent? Have you been to a nightclub or been to a place where they have UV light? Yeah? What glows more than anything? I'm hoping I'll glow today. That's why I wore this top. But white stuff, right? And when you watch a laundry detergent ad on the television, what do they tell you? It's their main thing. Other than getting it clean, what else do they want you to do? Or are telling you that their laundry detergent will do? Make your whites whiter, absolutely. Like they go on and on about it. So I have got, I'm not plugging these products, I'm just literally, this is what I had in my house. I've got some nappy sand, okay? That's the one in the pink bucket. I don't know, who does the laundry here? No one, none of you. It's mum's job. I've just got normal washing detergent in that one. And this one's Sard Wonder Soap. This promised me that it was going to make my whites the whitest that anything could ever make them, okay? So I'm banking on this one's probably going to be fluorescing a lot, Liz. All right. Shall we just give it a go? Yeah, I think we might. Let's turn the lights down and we'll see what happens. And we'll probably turn that away from the camera because... All right. Okay. Let's do those last because they are pretty cool. All right.
Do you want to do it?
You turn it on and I'll shine things through and hopefully people at home. All right. Okay. Laundry detergents. So Sard is absolutely glowing. The laundry detergent also really, really bright. How's that on the video, Steve? Yeah, it's good. And then the nappy sand, which I always assumed would make my whites whiter. There's got a few little molecules in there, but not, you know, a few little particles in there, but not a huge amount. In fact, it looks like stars glowing.
That suggests that they don't actually remove the stain. It just superimposes like fluorescence over so it looks clean.
It makes you white. Anyone use toothpaste that whitens their teeth? Just be aware because you will glow. Your smile will absolutely glow within a UV light. I don't know, do you use, we can check if you flick it up this way.
I don't think so.
Liz's teeth are glowing. I don't know if mine are.
Yeah, actually yours are, yes.
Yeah. So your teeth will glow because they've got whitening in them. All right, that's detergent. Money. We like money. All right. I don't know if you can, oh, my fingernails are glowing.
Oh yeah.
Can see the date. I don't know if you can see there in the audience. Steve's probably zooming in on there. You can see the date on there. Flip it over. And there's a bird on there that you couldn't see before. Visa card. It's got a secret V on there. I mean, these are detection systems of how we can detect fraud, just like your artwork. All right, remember what this was? Tonic water and olive oil. And you can see the tonic water is now glowing blue. And the olive oil is actually glowing red if you see it from the side view. Okay, the egg. Let me get it out of its amazing pink color. Okay, that's just a raw fresh egg out of the fridge. All right, over here, Liz, we've got, I'll move that out of the way. This one is just glowing. Interestingly, the pins, the orange ones and the yellow ones glow and the blue and the pink ones don't. It's just certain types of plastics fluoresce quite brightly. I know all Apple products glow brightly, you know, Apple pens, your earphones and things like that. This plastic is just taking off. Also, the white threads also glow heaps as well. Now this one, later on, you're welcome to come along and come down and have a really close look at this. The bananas is really difficult to see. I don't know if you can see that on the camera, but there's a little blue ring just around the dark spots on the banana skin, that's fluorescing as well. And they only obviously appear when the dark spots appear. So there's a molecule that's being formed there that will fluoresce. And then my pièce de résistance. So these flowers have been sitting in special fluorescing liquid, okay, that I've put in some water, and they've been sitting in that. I don't know if you've done the whole, you might have done this in primary school, put the flower in red ink or blue food colouring and it sucks it all up and it turns colour, yeah. So these are now fluorescing, and you can see it in the leaves and in the flowers along the veins. And again, I'm happy to, you know, you can take one home if you want, but it won't work unless you've got a black light. But they're pretty cool.
Yeah, I didn't know about the Visa card actually with the V. That's
yeah, no, I just pulled that out when I got here and thought, oh, I'll give that one a go as well. So just so many things will fluoresce, household materials. So there's that. We might turn that one off, Liz, and we're attached.
Oh, yeah.
And we might swap over to the other.
Okay.
All right, so we've got a whole heap of light globes here, fluorescing globes. Okay, ever wondered why they were called fluorescent tubes? All right. Do you want to explain this, Liz, or you want me to keep going?
No, you keep going.
Okay, so the top one, we've just got a fluorescent tube without the outer coating on it. Usually they come in white. In fact, all your lights now are probably being replaced by LEDs, okay? But these are the ones, and they used to flicker and cause all sorts of annoying things to our brains as well. But if I turn this on without the coating, it triggers it. What you're seeing here is UV light caused by mercury vapor inside the tube, okay? And when I turn the electricity on or the switch on, then that small charge here in the tube excites the mercury. And due to the thickness of the tube, of the glass of the tube, it blocks out a lot of the UV light, okay, which is good. But it's not producing a lot of light that we can see by, okay? So we put some coating on the light. Let me turn this one on. And you can see now this coating is really bright, right? And I don't know, is the video struggling to see this?
Yeah, it's a bit blurred out.
A bit blurred out. So on the left-hand side, we've got this coating that is really fluorescing. That material that's coating is really fluorescing and make it a lot more brighter. I'll turn that out. We've also got red coating and yellow coating, but we won't go into that one. The cool one is down here. We've got some more laundry detergent because, you know, laundry. We've got some just fluoro paper, okay, or cardboard, some of the glow-in-the-dark stars. But as Liz was talking about before, some minerals, which are really quite fascinating. And again, we can put this back on later, so you can have a look. So, but the tube down the bottom is a real black light, okay? It'll be producing UV light. And we can see all of a sudden everything's really glowing. We can see some of these rocks. Oh, they're all stuck in, aren't they?
Well, we can get them out.
There we go. That one's amazing. Really bright. Others, not so much. But yeah, some of them have got various minerals through them.
At the museum, they also have a display where you can turn on the light, and they've got all different types of colours there.
That's good.
Yeah.
Cool. Any questions? Might leave it loose and see if there are any questions on the chat.
Oh, yeah, you had a question?
What type of minerals do you have?
Oh,
No.
do you know what type of minerals
No,
there
not
are?
really.
No.
No, sorry.
But if you're interested, yeah, the museum has a massive display, and you go in and you can change the light source to see the fluorescent component of the minerals there.
What is coating the second tube like?
Yeah, do you know what the coating is here? It might be like titanium dioxide or something like this.
Yeah, maybe. I'm not sure.
It was phosphor on the
Phosphor?
Oh.
yeah, on the fluorescent tubes.
Yeah, okay, well, that one makes sense.
Different colour phosphors on these.
Right.
Okay.
And I think the washing powder's phosphor too, phosphor minerals
Oh,
ground
okay.
up in it as well.
Right. Yeah. Okay.
Okay. Yep.
Solar flowers, they weren't fluoresced by themselves.
No. So you've got to have that.
Don't be flowers in UV light.
What was the question?
Do bees see flowers
Oh,
in UV light, or do they see in UV colour?
I don't know actually about bees. I know there's like these shrimp that can see quite like low ultraviolet through to infrared. Like, but yeah, some flowers do fluoresce, I think, and like a lot of like certainly at the bottom of the sea, there's like an enormous amount of fluorescent like creatures, as well as all the coral fluoresces. Yeah, yeah. There's another example of luminescence here. Yeah, so in the second part of my talk, I was going to talk about fluorescence in biology, and this is more related to, I guess, like the research that I'm doing. So, but this is another example of how fluorescence is useful. So you saw before how we talked about fluorescence spectroscopy. Well, you can essentially perform this on a microscope as well. So rather than looking at a solution of a molecule that fluoresces and then recording its excitation and emission spectra, you can actually go in and image fluorescence as a function of space in a sample, and that's what a microscope offers. So here we have a laser scanning microscope, which essentially is what it sounds like. It has lasers that scan your sample in this raster pattern, and so in every pixel along this raster pattern, it excites fluorescence in your sample, detects the fluorescence, and then goes to the next one, excites, and then detects. And because we have this palette of fluorescent proteins, so I don't know how many of you are studying biology, but there's this, you might have heard of the green fluorescent protein. It was extracted from a jellyfish and has revolutionized like cell biology. And essentially it enables us to go in and tag any protein we're interested in with any color shown here, and then we can visualize that inside a living cell. So if you couple this technology with a laser scanning microscope that enables you to go in and sample fluorescence as a function of space, and you set up lasers and detectors using this principle of where does my fluorophore excite, where does it emit, you can individually go in and image and locate where your different fluorescent proteins are in a living cell. So if I had a sample that was stained with something that was fluorescing blue and it expressed something that's fluorescing green and red, I could set up a laser that excites the blue fluorescence, the green fluorescence, the red fluorescence, and then that signal would be sent up to where my detectors are. And by using different filters, like I did for the painting, I could direct the blue fluorescence into one channel, the green fluorescence into another channel, and the red fluorescence into a channel, and look at where these different fluorophores are located in my sample because of this raster scan on the microscope. So that's pretty good that we can do that spatially, but the nice thing about optical microscopy is it doesn't kill cells, so you can actually look at them as a function of time as they're alive. And if we set up temperature and CO2 control on the microscope, we can look at how, for example, a cell divides. So I don't know how familiar you are with cell division, but that's when the genome is essentially replicated to 2 daughter cells. And so, for example, here's a movie of a cell dividing, and you can see the DNA is labeled with a red fluorescent protein. And as it divides into two cells, you can monitor that as a function of time. And so here's another example. You can see the mitotic spindle, and then the cell divides into two. So just by labeling the different components of the cell, like the DNA that records the genome localization, and then the green fluorescent protein just is an inert protein showing you the cell plasma membrane, you can observe how, for example, DNA is dividing with quite high temporal resolution. So this is an application of this technology that's coupling the green fluorescent protein with laser scanning microscopy. So our question was, how does a cell detect DNA damage and repair it? So in every cell in your body, you have about 2 meters of DNA that's folded up into a volume that's only 10 microns in diameter, which is quite incredible. So how does the cell get all this DNA into your 10 micron nucleus? It folds it up into this very complicated structure that looks almost random, but it's definitely not. And essentially there's these repair proteins at all times scanning this really difficult chromatin network looking for DNA damage. So here's a schematic of, for example, where maybe a double-strand break has occurred in your cell, and the cell has recruited all these repair proteins to that site. And what people don't understand is how can all those repair proteins navigate this massive, like what looks like a mess of DNA, and know exactly where that DNA damage site is and fix it immediately. And what's been found is that the chromatin, or the DNA that's compacted into this structure, will open up at the double-strand break site so that these repair proteins are just naturally accumulating there and compact other regions of the cell so that they can't diffuse there. And so it looks like the DNA network itself is funneling these repair proteins to where they need to be to save the cell from this DNA damage. But the problem is, to observe this, the way DNA is packaged into what we call nucleosomes is well below the diffraction limit. So what do I mean by this? Well, in optical microscopy, there's this limit to what you can observe with light. And essentially it's around 260 nanometers in diameter. And the problem is these little structures that DNA is wrapped around in are only 2 to 11 nanometers. So how do we observe this? Well, fluorescence is really useful. There's this thing called photoresonance energy transfer. And so what is this? If we go back to our diagram where you learnt that if a molecule undergoes fluorescence, it'll absorb a photon and then emit a photon at some longer wavelength. Well, if there's another fluorescent molecule really close by that happens to absorb the same energy that was emitted by this fluorophore, so let's say we had some green fluorophore, it's excited at 500 nanometers, it emits a photon at 550 nanometers. If there's a red fluorophore really close by that happens to absorb where that green fluorophore emitted, then it will take that energy and emit a red photon. So there's a transfer of energy between these two molecules. And the really good thing about this in the case of biology is that the efficiency of this transfer process, it only occurs when the molecules are within 1 to 10 nanometers of each other. So essentially they have to be like really, really close. So coming back to our problem with the DNA and how it's organized, we could only see every 260 nanometers, but this event can only occur if the molecules are within 10 nanometers of each other. So if we label our DNA with green and red molecules, we'll be able to see where fret occurs, and if it occurs, we know they have to be within 10 nanometers of each other, and if it doesn't occur, then they must be more spaced. And so this is a way we can get around the diffraction limit and observe within 10 nanometers how the DNA is organized. So here we have a human cell expressing a green fluorescent protein and a red fluorescent protein that's tagging the DNA. And the idea is if the DNA is really compact, so like the repair factors can't access that part of the genome, you would see fret. But if the DNA was very open and the green and red fluorophores are very far apart, then you won't see fret. So I won't go over how we perform the analysis, but here is a human cell's DNA in green. And here's the cell where it's just normal. I haven't done anything to the DNA. This is how like it would regularly be. I then treat the cell with something that opens up the DNA into this more open state. And I've treated another cell with a drug that compacts all the DNA into this compact state. And you can see from the green fluorescence that they don't really look that different. They just all look like, well, to you probably just a circle, but like that's the genome organized into a nucleus, but they don't look significantly different. That's because of the diffraction limit. If we now go in and say, okay, but where's fret occurring in each pixel of these nuclei? You can see that the 3 nuclei are completely different. So when we look at the scale of nucleosome packaging, the red pixels show us where the nucleosomes are very close together, and the green pixels tell us where they're far apart. And you can see that in the untreated cell, you have red and green pixels, but if you treat the cell with a drug that opened up the chromatin, although it doesn't look very different from the point of view of the fluorescence, when you measure fret, you can see all the fret is gone, but when we compact it, you can see the fret is promoted. So now we have a way to look at chromatin on a scale that is relevant to the DNA damage response, even though the diffraction limit has prevented us so far from looking at this. So we have our way of detecting chromatin or DNA. So here's a cell, and I've gone with a laser and essentially burnt the cell in a way that I know produces a double-strand break. So I've cut the DNA with a specific laser that will induce this type of DNA damage, which occurs when you get sunburnt or any sort of exposure that induces DNA damage. And so I've cut the DNA here, and then I've followed that cell over 6 hours. And this is about how long it takes to repair a DNA damaged site. So here we now map the chromatin according to fret, like this 10 nanometer resolution. And if you zoom in on the damaged site, what you see is that the chromatin or the DNA at that location compacts, so it becomes very tight. And then if you zoom in on that region of interest, you can see that it opens up at a later time point, and the compaction is now around the DNA damage site. And what's interesting is if you follow a repair factor tagged to green fluorescent protein at the same time, what you see is that on this same time course, here's the DNA damage site I've induced, and you can see the repair factor is recruited to the DNA damage site as a function of time. But what was interesting is that when the chromatin is compact, the DNA repair factor is not recruited significantly, but as soon as it opens up, that's when you see the repair factor get accumulating at that site. So this is evidence that what we thought was happening biochemically is actually happening. When the chromatin opens up, that's when the double strand break site can be repaired by a repair protein. And essentially the repair protein never knows where it's going. It's the DNA opening and closing that funnels it to where it needs to be. And so this could only be observed by using this assay based on fret and the fluorescence from the fluorescent proteins that form that fret assay. So just to summarize, so now you know what is fluorescence. You've seen fluorescence here today. It's the emission of light from an atom or a molecule or any material that absorbs electromagnetic radiation. And it's informative for many areas. And I showed an example in the field of painting conservation, but I'm sure you're familiar with like forensics use it a lot because a lot of bodily fluids fluoresce, and also biomedical research, which I've shown here in the context of biology. With the advent of these genetically encoded fluorescent proteins and confocal laser scanning microscopes, we can more or less observe even the like as many colors as we want, really. We can label different parts of the genome, we can track proteins, we can differentiate cancer cells from non-cancerous cells just from their endogenous fluorescent signal. And so this is the example I showed was looking at quantitative imaging of nuclear architecture, and this all stems just from understanding the fluorescent process. And that's my email if you have any more questions. So do you have any questions?
I've got one. So you actually attach something to the DNA so it will fluoresce and follow it around.
Yes.
Yeah.
Yeah, and there's well, actually there's a lot of the cell has endogenous fluorescence. So what's quite interesting is, like, for example, cancer cells that have a very different metabolism to normal cells. There you don't have to attach anything. The cancer cells have a different fluorescent signature and you can just image endogenous fluorescence. But most of the case, if you are interested in a specific protein or a specific structure, you need to label it.
But you can't put like a body underneath your fluorescing machine and map out the cancer? Is that
It's like becoming maybe like yeah. So there's it's becoming maybe an area, particularly like in the context of skin cancer. But yeah, it's still, people still want a biopsy because at the end of the day, you'd like want to know for sure. Like it's pretty good, but maybe it's not as accurate. But for biomedical research, it's good for understanding the metabolism of cancer. But it's also, yeah, like prosciutto also has an endogenous fluorescence signature, and you can date the quality, or like figure out the quality of the prosciutto from the fluorescence. So they use it there, but yeah, it's still, yeah.
Yeah. You mentioned earlier that fluorescence is from different subshells. I know that, for example, the 2p and the 2s orbitals are different energy levels. Would you be able to have fluorescence going from different subshells like 2p and 2s?
You can have like there's only, it depends. You can have what's called like intersystem crossing, and so for fluorescence, for example, everything's within the singlet state, but if you have phosphorescence, it transfers into this triplet state, and that's why it lasts longer. It depends on the fluorescence lifetime of the molecule. So I'm not sure if that answers your question, but it's also like a probability in some way. So it's not as like you think there's like a weighted probability towards how the decay will take place. But yeah, I don't know if that answers your question.
I mean, theoretically,
yes. Yeah. Yeah, I think
you can go from
any, yeah. But like in general, like in the condensed state, things will always relax to that ground state vibrationally, or in the context of a molecule though, to that first excited state, and then the photon is emitted from there.
And that's why we shine UV light on those objects, but they fluoresce invisible, like we can see the visible light coming off them.
Yeah, because you've lost energy due to the vibrational relaxation. Yeah.
So what's the difference between the light at the top and the black light at the bottom?
So apparently the glass at the black light at the bottom isn't blocking as much of the UV component, so you can see the purple emission coming through, whereas like in a lot of windows, they block UV light. So like when you're driving, so you don't get sunburnt, and there's a protective quality because UV is like, it's bad. In fact, UV is what you use to induce DNA damage in those experiments. Yeah. But it's great for fluorescence.
Yeah.
It excites more or less everything. Like in those pigments I showed you, they all had like a small absorption in the UV region in addition to their daylight fluorescent component.
There was also that suntan when they had like the black, and then they put the black light on, and you can see everyone fluorescing and all the sun outside and everything.
Yeah, like, and I think in some sunscreens there's like UV absorbers that were potentially causing some issues, and then other sunscreens they're beneficial. So I don't know.